let's move on to the morning's last presentation in the health area, the Keynote 3. As for the previous sessions, do not forget that you can follow the conference's live streaming on the IPB channel on YouTube, and use chat to place your questions. I remind you that you should send your questions during the presentation and do not save them to the end, as we may not receive them on time, and therefore we will not present those questions to the speaker. So Keynote 3 will be presented by Dr. Adelar Bracht from Maringá State University, who I am pleased to present next. Dr. Adelar holds a PhD in Natural Sciences from the University of Munich and is currently a senior professor of biochemistry at the State University of Maringá. He has been doing research work since 1980, focusing on liver metabolism, including oxidative stress. I welcome Dr. Adelar and invite him to present his keynote entitled The Adjuvant-Induced Arthritis Model and its Utility for Investigating Anti-Inflammatory and Antioxidant Effects. Dr. Adelar, welcome, and please, you have the floor.
Thank you. You are hearing me?
Yes, perfectly.
Yes.
Okay. So, let's move to the next. So there are many in vitro systems that allow a preliminary evaluation of these properties. In general, however, these characterizations are relatively uncertain because they are artificial conditions, which mostly have little or no resemblance to the in vivo conditions. Especially the effective doses and concentrations are frequently uncertain. These are the main reasons why in vivo models are in fact needed. These models represent an intermediate step that precedes the final goal to be reached, the clinical trial. The adjuvant-induced arthritic rats present several favorable features for being used as an in vivo model. First of all, several inflammatory, oxidative, and metabolic indicators are modified. Modifications are essential. This is because it is extremely difficult to improve conditions that are already close to normal. The modifications in these rats, these arthritic rats, are not restricted to the joints, but they usually become systemic and affect a lot of organs and tissues. The modifications in these rats, furthermore, are very similar to the modifications that occur in rheumatoid arthritis. This is important because this generates a translation factor. So you can translate eventually these results to rheumatoid arthritis patients. Because in patients, in the actual patients, limited interventions are possible. You can obtain, for example, blood samples, or eventually samples from other tissues. In the rat, however, all organs can be analyzed. So you will have a parameter diversification, which increases certainly the importance of the results you are obtaining. In the rat, arthritis can be induced, for example, by injecting the so-called adjuvant, which may consist of HIV inactivated Mycobacterium tuberculosis, for example. If this adjuvant is injected, for example, in the left paw, and edema developed quite rapidly, which is evidenced by this pore volume increase that is observed here. Later on, the non-injected pore, in this case the right hind pore, also develops these lesions. And in time, the disease becomes systemic. It extends to the whole body, so that practically all organs, or at least most important organs, are in fact affected by the disease. This produces many changes that can be measured. These changes should be sensitive to anti-inflammatories or antioxidant agents. Here we see what happens in the liver, for example. This work was published a few years ago, in which we analyzed the changes in the liver cells. This work specifically, but the scheme bases on several modifications that were detected by preceding work. As you can see, circulating cytokines induce several transformations, which include, for example, increased glycolysis because glucokinase is increased, decreased glucose 6-phosphatase, which is in line with decreased gluconeogenesis, decreases in the NAD/pH NAD ratio, which is connected with the decrease in GSSG ratio. And all this leads to several modifications, which include increase in reactive oxygen species, which means increase in protein oxidation, increase in lipid peroxidation, and so on. There is also an increase in nitric oxide synthase, decrease in antioxidant enzymes, and so on. All these parameters can be measured. And potentially, antioxidants, natural antioxidants, should affect these parameters. That is an important fact that I would like to emphasize here. These effects are compartmentalized. You see here 3 compartments. We have distinguished these compartments: the cytosol, peroxisomes, and mitochondria. The effects may be different in these 3 compartments. Ideally, one should not analyze the whole cell contents, but would be more appropriate to analyze different compartments, although this is linked to hard work and is rarely done, in fact. So this gives us a general picture of what happens in the liver. You see, all these parameters can be measured or should be measured when testing and the antioxidant effect of a given compound. Similar things happen in the cardiomyocyte. We have published this paper a few years ago too. The effects are equally compartmentalized and may even be different depending on the parameter. For example, thiols are increased in the endoplasmic reticulum, but decreased in the endoplasmic reticulum, but increased in the mitochondria. The extent of the effects may be different, but in general, the same parameters that are modified in the liver are also modified in the heart. A similar picture we have in the brain. This is a neuron, and we show in this picture the main modifications that may occur when the rat becomes arthritic. Now, arthritis induces a series of modifications in the brain, as you can see, which are similar to those that occur in the liver and in the heart. But here we have also measured the transmembrane potential of the mitochondria, which is increased, a fact that certainly contributes to the increased production of reactive oxygen species, which lead to lipid peroxidation, protein oxidation, modifications in the level of GSH, which is a decrease in this case, and modifications in the nitric oxide concentration. The mechanisms, the basic mechanisms, are always the same. There is an increase also in xanthine oxidase, which is very pronounced in the brain, and which is a parameter that can be measured when you are investigating the effects of natural compounds on the oxidative status of these rats. Well, these are the main changes that occur and that can be explored when you are measuring the effects of natural compounds. We have done this with a series of natural products, purified or not. We have done this, we have investigated the action of several extracts with generally convincing results. I am showing here, before I show some of our results, another picture about the liver, which shows modifications in metabolism. Arthritis modifies metabolism in several ways. As I already said, glycolysis is increased, gluconeogenesis is decreased, respiration is increased. This is generally coupled to an increase also in the oxidation of fatty acids, as shown by this work that we have published more recently. The mechanism is always the same. By the way, adipose tissue is equally affected. We have not investigated adipose tissue, but it is equally affected. So you have an increase in the oxidation of fatty acids, which leads to an increase in production of CO2. In consequence, you have an increase in the respiratory chain, which in the brain at least also reflects an increase in the mitochondrial membrane potential. Changes in the NAD ratios, and so on. These are also parameters that can be measured, and it would be highly interesting to know if natural antioxidants and anti-inflammatories really affect these metabolic parameters and not only oxidative stress indicators. Of course, there is an increase in reactive oxygen species and so on. But things are correlated. There is an intense cross-talking. So it would be interesting to investigate metabolic parameters as well. Next, I will show you, I have not so much time, of course, I have wasted some part of my time in trying to present this speech because I am an old-fashioned man. So I would like to show you results that were obtained with two different agents. An extract, which is an extract of Ilex paraguayensis, which in English is normally called yerba mate, using this Spanish word in Portuguese, erva mate, or other designations here in Portuguese and Spanish, which is an herb which is consumed in southern Brazil, in Argentina, Paraguay, and Uruguay. In this way, these girls are drinking what is in fact a hot water aqueous extract of this herb. The herb is dried, still green, and it is used to prepare this beverage here. It is very rich in methylxanthines, which is caffeine and theobromine, for example. It is highly stimulating, but it also has other effects. In our study, we used aqueous extracts mimicking the way by which the herb is traditionally consumed. For example, these are measurements in the plasma. These are plasma parameters. I have selected some parameters. I'm not showing the entire work, which has been published in Food and Function 2 years ago. This work was done in collaboration with the Mountain Research Center, which determined the content in phenolic compounds of the extracts that we use. You see here, it is important to use more than one dose of the drug. It is important to use appropriate controls, which is the normal rat, which was also treated with 2 doses of the extract. The arthritic rat presents increased plasma protein carbonyls, which measures the lesion to proteins, which a parameter measures lesion to proteins. There is also an increase. Sim?
Não faz mal. Estive a falar com a professora e por acaso estava a falar com ela porque tive outros problemas hoje de manhã.
I'm so sorry.
So, arthritic rats present increased myeloperoxidase levels, increased plasma, it decreases the thiol levels in the plasma, and decreases FRAP reactivity in the plasma, which is a measure, can be a measure of antioxidant capacity. As you can see, treatment attenuates these effects to some extent, sometimes more, sometimes less. The effects are not always that pronounced, but there is a clear effect, attenuating effect. This is an anti-inflammatory, an inflammatory indicator. This is an oxidative stress indicator, an oxidative stress indicator, and also an oxidative stress indicator. The effects are different. The extents are different. This cannot be predicted from in vitro experiments. Similar results were obtained when the liver was investigated, as you can see here. Protein carbonyls are increased in the liver as well as in the brain. ROS levels are increased in the liver as well in the brain, and treatment with the extract decreases these levels. It does not really normalize these levels, but this certainly and apparently increasing the dose will not increase the effect, but there is an effect, a decreasing effect, normalizing tendency when rats, arthritic rats, are treated with these extracts. GSH, glutathione, reduced glutathione, is not really much increased than normal rats are treated with the extract, but they are decreased by arthritis, and there is a partial recovery, as you can see. But the effect is much more pronounced in the GSH-GSSG ratio, which is actually a redox ratio, and which is much more important. This occurs in both liver and brain. So what I want to conclude. I want to conclude about this few results that I showed to you, that you can indeed, in the arthritic rat, measure and detect antioxidant and anti-inflammatory effects, which are predicted by in vitro measurements. But the in vitro measurements are not able to predict the extent and the doses that are required to produce these effects. And there are also other parameters that are not affected. They should be affected, but they in fact are not affected. I have not enough space to show all these results, but I can assure you many parameters are simply not affected in spite of the intense antioxidant activity of the extract. Ideally, to finish, ideally, one should always use purified compounds. In the case of the yerba mate, Ilex paraguayensis, there are no purified compounds available, or most of them are not available. Chlorogenic acids, you generally cannot get great amounts of these compounds. But there are other compounds that are available, readily commercially available, as for example, resveratrol, which, as you all know, is quite abundant in grapes, for example, and in many other fruits or products of plant origin. Well, resveratrol is a panacea. Many effects have been attributed to it. What interests us is the anti-inflammatory and antioxidant effects. And we have proposed recently, have done recently a work in which we have changed our mode of doing experiments with natural products. Instead of using a single dose or two different doses, a low dose and a high dose, we have used various doses, several doses. We have done a multiple dose investigation for several reasons. Multiple dose investigation, although the committees, the ethic committees do not like, because you must use many rats, many animals for your study, I think it is important because it can bring to light many different effects and particularities of the actions of various compounds. This work, which was recently published, shows that resveratrol inhibits, in fact, the production of reactive oxygen species concentration-dependent manner in isolated mitochondria. These mitochondria were isolated from the brain, where these are isolated mitochondria, so that all factors have been eliminated, all factors that might be increased production of reactive oxygen species in vivo have been eliminated. Even so, this is important, in my opinion, to investigate the action of natural products in isolated mitochondria because you may be surprised, and we have been surprised in several ways, in several occasions, because sometimes the compound may act as a pro-oxidant, in fact, and not as an antioxidant. Well, here are some results that I have picked to demonstrate the action of multiple doses of resveratrol, a clear concentration-dependent effect on the myeloperoxidase activity in plasma. As you can see, in arthritic rats, it decreases the activity. No effect, or no clear effect in control rats, but resveratrol was not able to modify the diminished plasma albumin concentration. And the effect in control rats is a mess, difficult to interpret. But there was a decrease in the increased xanthine oxidase activity. But here again, the effect in control rats was a mess, no defined effect, a declining tendency here, but this is difficult to interpret, a almost normalization of the effects, of the negative effects of arthritis in catalase. Dose dependent, of course. And I would like to emphasize, before I end my presentation, the effects on plasma thiol concentration, which is illustrative of the reason why we should do multiple dose experiments. As you can see, arthritic rats present low concentrations of plasma thiols. This is consistent with oxidative stress. And this is mainly due to the low levels of albumin, of plasma albumin, as plasma albumin is the source of most thiol, reduced thiol groups. Well, the various doses of resveratrol had no effect on the thiol group concentration in plasma. In the opposite, they had an increasing effect on the plasma thiol content in healthy rats. This is antioxidant effect. The plasma albumin concentration was not changing, in fact, but its ability to have antioxidant effect has increased when resveratrol was given in a concentration-dependent manner, but there is a maximum here and a decline afterwards. So actually, this shows that resveratrol has two kinds of effects: antioxidant effect at low doses, and later on, a pro-oxidant effect. This appears quite clearly in healthy rats, but nothing happened in arthritic rats. But here another factor appears, another factor intervenes. In arthritic animals and patients, there are higher levels of glyoxal and methyl glycol. These compounds tend to promote resveratrol-induced oxidation, even at low doses. So actually, the interaction of resveratrol with glyoxal and methyl glyoxal is actually impairs the possible antioxidant effect of resveratrol on plasma albumin. In conclusion, you can conclude that all these things can in fact not be predicted by in vitro experiments, actually not even in cell systems. You have to use animals, unfortunately, to discover or to see experimentally all these different effects. So let me conclude. So in our experience, the adjuvant-induced arthritic rat is an appropriate and convenient model for investigating anti-inflammatory antioxidant effects. Advantages are similarity to the human disease, a fair number of parameters can be measured, several and important organs are affected, high reproducibility, pro-oxidant actions can eventually be detected. For the future, multiple dose experiments are recommendable for increasing both significance and validity. Effects on the metabolic parameters changed by inflammation and arthritis may produce new insights into the mechanisms underlying the disease. Thank you for your attention. I'm sorry for my lack of training in this kind of presentation. At the beginning.
So much.
At the beginning.
It happens all the time. So thank you, Professor Adelar. I don't know, I guess our audience is very shy and they are not questioned so much.
Thanks God.
No, but thank you for your interesting and clear presentation. And thank you for meeting the allocated time, even with those problems at the beginning. Okay, I think we are not receiving anything. So I just have one or two curiosities. They are not questions. We saw in your presentation, of course, and we know that you tested not only yerba mate or resveratrol. So when you tested some of the natural compounds that you choose, did you test them, all of them in their free form, like this aqueous extract, or did you isolate some compounds, or you have only results for steel beans like resveratrol? You tested mostly extracts in their free form?
To now, most of our works, our studies have been done with extracts, but always characterized in terms of their contents.
Okay.
Characterization, as I don't know if I mentioned it, is generally done at the Mountain Research Center. It's a collaboration that we have with this highly productive group. But we have also done several studies with isolated compounds. I gave you this example, resveratrol, and we have several other studies in which we use the purified compounds.
Okay. So
the trend, of course, is to use purified compounds, so you can really know what compound exactly is doing what. This is very, very important, I think. But sometimes it is difficult to obtain the main products that exist in extract.
Yeah, of course. So gathering all the data obtained in your work regarding the natural compounds that have shown promising effects, in this inflammatory process inherent to the rheumatoid arthritis disease, you cannot establish yet a relationship between the obtained results and, for example, the compounds present in these extracts, and infer about the most promising classes of compounds, right? We don't have those answers.
Yes, you have a group of compounds, but you can't say with certainty what exactly compound is responsible for what, as I emphasized.
Okay, of course.
You can translate the doses. Look at Ilex paraguayensis, there is a lot of consumption. There are people that drink these infusions during the whole day. So they consume a lot of these compounds. You can make translations because they are formulas to the human case, to the human situation, I mean. You can make translations using certain formulas, of course. But it would be highly desirable if all these compounds were available in pure form. But you need sufficient amounts. Because in vivo experiments consume much greater amounts of specific drugs. In vitro, you have very, very small volumes, and you can test at will. You can repeat tests and so on. This is a disadvantage of in vivo experiments.
Of course.
But you can use very small animals. Instead of rat, you can also use mice, which are much smaller and require much smaller doses.
Okay, thank you. Okay, I guess the participants have hungry. They want to have lunch. So I think I will end the morning session that had as a theme the application of natural products in the health area. I thank all the speakers who presented us with their communications, and I also thank the entire audience for having accompanied us and for your participation. Any question or doubt, contact us through the links available on the Congress website, email, and social networks. Before I leave, I have just one message for André Folgado, one of our speakers in this morning speech session. Since there were problems during your presentation, André, we were unable to view your slides. So if you want, you can make your presentation at the end of the day, at the end of the last pitch session, okay? You can contact us for any further information. So thank you, Professor Adelar. Thank you all the speakers in this morning session. So I declare the session closed. We restart at 2:30 p.m. With a session of natural products applied to the food sector. Have a good lunch, everyone. See you in the afternoon at 2:30, okay? Bye-bye.